All right, so what we're going to do today, this will be a short and easy class. We're just going to warm up. You've all been away on spring break, right? Did anybody do anything exciting?
Catch some jitters.
What did you do?
Live by the pool, loving, the usual college kids.
That sounds delightful. Yes. Yeah. I did the usual old college professor thing of visiting my grandchild. So that was nice too. But otherwise it was staying home and watching the snow melt. So we've got spring break out of the way. The first thing we got to talk about is the lab. So where are we at with the lab? So some of you I know managed to get white miniature forked by wild type crosses going before you left, right? Okay, good, you got that. Anyone else get that far? If you didn't, it's okay. So what we need you to do first thing this week is everybody needs to stop by the lab and kill flies. Just clear them all out. And we'll look for fresh flies emerging, and those are the ones we're going to use for the next cross. So some of you have white miniature fork bottles and you have wild type bottles, but they weren't producing before spring break. Go in today or tomorrow and clear them and go about collecting virgins for this next cross. So that you're going to cross white miniature forked by wild type. We're going to get hybrids out of this, and then we're going to do a test cross. Some of you already have those flies done. That cross is ready to go. And with any luck, you'll go into the lab this week and you'll already have flies emerging, ready to go. You're going to clear the first batch, clear the parents, and then you're going to cross the hybrids by the hybrids. In lab tomorrow and Thursday, my plan is just to have a grand assessment. So we'll all just sit down and see where each group is at and make an individualized plan for what you're going to do to get it all done. Okay, so that's in progress. The other thing is we've still got to finish writing up our lab report for the last lab. And you guys got the methods done, you got the introduction done. You're going to do the results this week. So I will post an assignment for the results with some specific requirements. Your results are going to contain a couple of tables. You can predict that. One of the things I want you to do is crunch the data a bit. So right now we've got all these different groups in columns. We don't care about individual group results. All that matters is the total. So for instance, your table, one of your tables will be just the raw data. And then you're going to have to do some simple analysis on that. Okay. That experiment is all done except for the write-up. So the plan is you're going to write that up, the results this week, the discussion the week after that, and then turn in the final paper for the complementation cross. Then I'll grade that, get it back to you. If you need to, you can do a revision. So if I notice some terrible problems in that draft, yes, you can do one revision to get your grade up there. Then we're going to be working on the second cross, the mapping cross. We're going to do that one a little differently. I'm not going to hold your hand anymore. So I'm not going to have you turn in each chunk step by step, because you know what you're doing now, right? You're supposed to say, yes, we know exactly what we're doing now. And so the write-up for the mapping cross will just be one big paper you turn in. Again, I'll take a draft. I'll give you a chance to revise it if there are problems, but we won't have this incremental thing going on. All right, what else do I need to say about the lab? Everyone feel like they're under control? They know what they're doing, sort of, more or less? If you don't, we will fine-tune it and get it all explained in lab this week. All right. Now, what I'm going to talk about today, I'm just going to give you a couple of problems to get your brain back in gear, thinking about these things, a few triple point cross problems, and then I'll let you go. There'll be a hint. There's going to be another batch of problems assigned for the weekend, so we'll do that. Also, reading your exam, the exam 2, a lot of people made suggestions about where they were feeling weak. And I know people want me to go over sex linkage again. So we might do that Wednesday. But I also want to move on to chapter 8, which is chromosome changes and things like that. So we'll see. I'll leave it open a little bit. I know this is a non-representative chunk of the class, but do you guys want me to go over sex linkage again on Wednesday? Somebody shook their head. No, you're tired of that. Yeah, you know it all. Okay. Well, we'll see. If I get comments that say, please do, I will do it. Okay, so where we're at. Again, this will be a reminder, I hope, that we did a bunch of triple point problems before spring break. You remember that? Now, you've been sitting on the beach. It's just cooked out of your head.
There's no beaches in Phoenix.
Oh, you were in Phoenix. Oh, but there's sunshine.
Lots of that.
Yeah. Okay, so the brain bleached. It was by the sun, so we'll see if it all comes back to you. Okay. Program. Oh, look, did I turn that on? Come on. You can do it. Oh, no, I don't. I forgot my little widget. Okay. We'll do it by hand. All right. So, look at all those numbers. So this is a set of typical triple point cross numbers. I got 5 data sets up there. Each one of those data sets is independent of all the others. So we'll just look at them one at a time. And the first thing I'm going to ask you to do is a really simple logic problem, which you should have mastered by now. That is, let's just look at one for a moment. This column right here. And I tell you, okay, these are the numbers we observed. What is the order of genes on the chromosome? Alternatively, you could think of, all you got to do is tell me which one is in the middle. Is A or B or C in the middle? Have you already got the answer?
I think so.
Oh.
I think A's in the middle.
You are correct. Oh man, this is going to be a short class at this rate. Yeah, this should be, like it says here, I want you to do this just by inspection. You don't have to do any fancy calculations. Just got to deduce the logic here and figure out which gene is in the middle. So you glanced at this and just figured, oh, super genius is good. So how'd you do it?
So we know that the all wild type and then the A, B, C at the top and bottom, those are the double crossover. Or no, the non-crossover.
Non-crossover, yes.
And then the 2 in the way middle, yeah, those 2 are the double crossovers.
Very good.
So then
yes.
you just figure out which one's the odd one out.
Okay, so for instance, you look at this one and compare it to that one, and you see, well, these 2 are different, and this one is the same. So it must be that letter A must be the one in the middle. Does everyone see that? So you can just do a quick comparison. You should be able to do these just by glancing at them, figuring out who's non-crossover, who's single crossover, or double crossover, and just do the comparison. So I got the answers here. Let's see. Oh, yes. Good work. What about the second one? Same method applies. Go ahead.
C would be in the middle?
C would be in the middle. How did you get that?
Because with the 1 and 1 really tell anything, but with 4 and 4, C is the only one that's really changed between the two of them.
Wait a minute. Which one?
Oh,
These two are the double crossovers, right?
yeah.
So you know this is a double crossover. And then you know the non-crossovers have got to be these two. And then you compare this to, for instance, this one, or this one to this one. Yes, it would be A. Your logic is right. You just fluffed it on the last bit. But yeah, you compare that to that, and that tells you that A is the different one. So A is in the middle. Another important thing when you're doing this is, like I said, the first thing you got to do, who are the non-crossovers? Who are the double crossovers? Make sure that you identify the 2 non-crossovers and the 2 double crossovers, and then make sure they complement each other. So like here, A, B, C, and plus, 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 yeah, they complement each other. And 77 for this, that's plus B, C, and A plus plus. Those complement each other, so that's the correct double crossover and non-crossover types. Okay, the third one. Yes.
I got A again.
You got A again. Means just A. Oh, I should have. Yes, that's A. And the third one is A. Man, that's boring. Okay, how do you figure that one out?
So it's going to be the 6 and 3 are the double crossovers.
Okay, right there. All right.
And then the 339 and 291 are the non-crossovers.
And those two, okay.
And then the odd one between those is A.
All right. Yeah, so let's do a little quick comparison. So if we compare these two, then you can see, is A the odd one out? A is the same in both of those, and the other two are both different. Yeah? That works. Yes, it's A. All right, number 4. You think you'll be A again? You've already got it?
That one would be C.
That one would be C. Yes, you are correct. Yeah, I had to break the streak there. Once again, the logic behind this, how do you figure that one out? This one has some funny stuff going on in it, because look here, there's 4 that are fairly numerous, and they can fool you. So which ones are the non-crossovers?
235 and 232.
You're saying this one and this one. Going to double check, do they complement? And that's plus plus C, and 235 is AB plus. Yeah, those complement each other, so those are probably the non-crossovers. Also, if they're really close, add them up. And the one where the sum is the largest is the one that is the non-crossover. All right, and then the single crossovers are?
46 and 48.
Yeah, that's plus plus plus. And if we compare that to this one, for instance, which one's odd? The C. So that's the answer there. Okay, last column. Somebody who hasn't answered before. Let's see you exercise your brain. Again, these are nice because they don't require any math skills. They don't have doing fancy stuff. It's just pure logic, looking at the arrangement and the numbers. Yes?
Is it C?
It is C again. How did you get that?
So 0 and 1 would be the single cross, and 300
Double crossovers.
Double cross,
Yes.
sorry. And 305 and 265 are the non-cross.
So
the different one would be C.
that's a non-crossover, and this is a double crossover. And again, you compare those, it's got to be C, that's the odd one. Okay. See, that was easy. I'm breaking you in gently. Now let's go a little further. So what I'm going to do next is we are going to look at just this set of data right there, that data set number 1. And we're going to figure out the map and all that kind of good stuff. So we're just going to do that, and here's the warning for the assignment for this week. Part of the assignment this week is I'm going to give you the other 4 and tell you to do the same thing, figure out the actual distances. Okay. So we're only using data set 1, and I've copied it right here. Here it is. You already figured that. I even tell you which one was in the middle, because you already figured that out too. And I say, okay, first thing to do is to organize the data into this table. This is kind of a handy table because it will allow you to quickly puzzle out what's what. Now, once again, we're going to say, okay, we got to go for the non-crossover category. Which ones are the non-crossovers?
317, 203.
That one and that one. So we can put those phenotypes over there. And one thing I did here is I rewrote them in the order. We know that A is in the middle. That's why it's kind of handy to first just glance at the data and say, I know which one's in the middle. Because then you can reorganize the data as you go. And the total there is we've got 203 plus 317. We got 520 in that category. Okay, next, double crossovers. Let's do the double crossovers. Which ones are those? Yeah.
B plus C and plus AT plus AC.
Yeah, these two. And again, you've already done it naturally. You just reorganize, put them in the A in the middle, right? So when we do that, we get, there's B plus C and there's A plus A plus. And we can also get those numbers. There's just 2 of them total. Okay. Next, we want to figure out the single crossovers. So it doesn't matter which order you do them. Let's just start with the biggest single crossover category, which would be this one and this one, right? So we take those 2, and that's plus C, which, and this one is BA plus. And so we work those out, and we add up those numbers. That's 130 of those. Now the other thing we can do next is we can say, okay, this is a single crossover. There are 2 single crossover categories. This is a single crossover between which 2? And to get that, you just compare one of these to one of the non-crossovers. Yeah?
Would it be between A and C?
This one would be... yeah, so that's what we got. Wait, did I get that? Yeah, that's correct. So that's a single crossover between A and C. Then we do the other single crossover. And that's going to be the leftover ones. That's going to be B++, and that would be this one, right? Yeah. Okay, so we get those in there. And the numbers of those, again, we got small number 31. And we know that A is in the middle. So this one must be a single crossover between A and B. So we get that in there. Okay. What do we do next? Well, we got to figure out the frequencies. The raw numbers don't tell you much until you calculate the relative frequencies. And so, for instance, we can add those up. There's 683 total here. What a disgrace. I didn't come up with a nice round number for you. So there's 683 there, and then we can ask the frequency of each of these categories. Each of these 4 categories. So you get the principle here. You're generally going to be breaking things up into 4 categories. The non-crossover, the double crossovers, and the 2 single crossover categories. And you get 76.1% for the non-crossovers. You get 19% for crossovers between A and C, which tells you that A and C are roughly 19 units apart. 4.54 for the A and B distance. And then very important, you got to get that double crossover frequency. So we get 0.029 there. And to get the actual distances, then what we have to do is add this number to that number, and this number to that number. So we can get a map that kind of looks like that. Someone see how I made that? Again, we haven't anchored either end of this, so it can be any order. You could have done it C to A to B. That works just fine. So we get a nice little map here. Is it all flooding back? Do you all remember how to do these? I'm sure you do. Yeah, okay. So, here's another one. And we're going to do this one a little differently. So I want y'all to pull out a piece of paper and just do this calculation all on your own, or you can work with your friends. That also works. That's fine. But give me the answers to those questions. And I'll just sit back and take a nap while you're working that out. I won't really take a nap. If you have questions, fire away. But this is a problem just like that last one.
Do you have any hints as to what the expected DCO would be?
What do you expect? Oh, you figured that out from the map. So remember, you do the map, and then you find the distances between some of the genes. And then the expected DCO is obtained by multiplying those frequencies together. That's the probability of having them cross over in both places simultaneously. You must be working on E. Very good. Okay, are we ready to start telling me the answers? Oh, we'll see. Okay, the first question, what is the arrangement of alleles in the female parent? And how do you figure that out? Again, this one doesn't require any math, no calculations involved. Are you raising your hand very tentatively? Go ahead.
BL plus, PR plus, and L plus.
Oh, but that isn't the arrangement of the alleles. That's the genotype. The female fly is heterozygous. But when we say the arrangement of alleles, we want to know what's the arrangement on each of the 2 chromosomes. Anyone else? Go ahead.
BL, PR, L. Yeah.
That would be 1 chromosome, but it's not the correct arrangement.
Oh, wait, okay, hold on. BL, PR, L.
No. I think you're trying to give the order, but it's not. The order doesn't matter here. How do you get this this arrangement of alleles, it's really easy. You just look at the non-crossover phenotypes. So this one and this one, right? And that tells you that on one chromosome, she's got PR++, and on the other chromosome, she's got plus BLL. If I draw it out. So that's the arrangement of the alleles. So you see the mother was heterozygous, but her parents weren't. So we got to figure out what was the arrangement of alleles in her parents. So this female was produced by a cross of a BLL fly by a PR fly to produce this heterozygote. Okay? Yes?
It doesn't matter the arrangement.
At this point, we don't care about the arrangement. When I ask for the order. Yeah, at this point, when I ask for the arrangement of alleles, I just want to know that you could then figure out how this cross produced this female fly. Okay, B is an easy one, right? What's the genotype of the male fly?
All the small ones.
All the small ones.
No pluses.
Well, but wild type is recessive to low.
Oh, okay. BLPR plus.
Yes, BLPR plus on both chromosomes. He's homozygous recessive. Okay. Yep. And then we ask which gene is in the middle. We did this a whole bunch of times just a moment ago. I'm sure you can figure it out. Again, we're looking at comparing non-crossover and double crossovers. Yes? PR. Very good. Yeah, so you can, for instance, just compare this one to this one, and you see PR kind of stands out there. So PR is the one in the middle. So up there, I could go back and rewrite this and say, oh, well, the arrangement of alleles in the female parents is BL plus L, and then plus PR plus. But it doesn't make any difference in this case. All right, draw a map of the three genes. What kind of distances did you get? Go ahead.
I got 5.2 between BL and PR, and then 16.7 between PR and L.
That sounds in the ballpark. Okay, here I've thrown the whole thing out here. Did you say 3.2 and 16?
I said 5.2, but maybe I did my math.
Uh-oh. Yeah, it's got to be 3.2.
Yeah, I must have done it wrong.
Okay.
Just off by 400.
Yeah, just off by about 40%.
Yeah.
Okay, so anyway, what I did here is I just threw it all into a little arrangement. You went with 4 rows for the 4 categories, just like I showed you in the previous examples, and then just organized everything. So the non-crossovers have to be BL plus L and plus PR plus. And the total number is there, 7,820. Then I work out the single crossovers and figure they're a single crossover of what? This is between BL and PR, and this one's between PR and L. And there's the numbers there. There's the double crossover frequencies. And then you get these numbers for the distances, 0.166 and 0.031. You add 0.001 to those. So this is 0.032 times 100 gives you 3.2 and 16.7. So does that all look right to everyone? Is that what you guys got? Okay, coefficient of interference. I just went ahead and threw it on here. Coefficient of interference is going to be determined by looking at the expected frequency of double crossovers, which is going to be 0.167 times 0.032. Okay, yes?
Are we sure it's not the 5.2? Because I redid the math.
Oh, you did? You got the same thing? What did I do wrong?
Because you add the 260 plus the 250, so that would be 510.
Okay. Yeah, so how did I get, I'm the one who screwed up. Yeah, looking at those numbers, you are exactly right. Okay. I was the one that was off by 40%. But you are correct. It should be 5.2 right there. So I calculated the coefficient of interference by taking 0.032 times 0.167, and I get 0.05. 0.005 double crossovers were expected, and we got 0.001. When you use 5.2, that should be 0.052. What's the expected frequency of double crossovers then?
0.008 or 0.009.
0.009. And we observed, wait, is that right? 0.009 or 0.09?
Yeah, 0.86.
Wait, we got to worry about the decimal point here. So, let's see, I will. No. This is really annoying. That face recognition doesn't work when you wear these things. Have you ever noticed that? Okay, so we got 0.167 times 0.052. 0.008. Okay. We'll round up 0.009. You're right. So we got 0.009, and we expected 0.167. So we divide that by 0.167. And then we get 0.052. So 1 minus that. So we got, you've all done the calculations. I will fix this before I post it on the web. And yeah, so we can calculate the coefficient of interference with this. 1 minus 0.052. So, which makes sense. So we're using a larger number than I put there, so it's going to, for this. So yeah, we got like 95% interference in this experiment. We got a lot fewer double crossovers than we expected. Okay, so that's all I set up for today. We'll go through a few problems. Your brains are all warmed up, right? Y'all remember this stuff. So for Wednesday, start reading chapter 8. This is the fun chapter. We talk about chromosomal rearrangements and how things get messed up during meiosis. And we'll also have a little bit to make the pre-meds happy, because we'll talk about some human syndromes that result from chromosomal disorders. Okay, we'll see you in lab.